Namaste to all my lovely students. Welcome back to the channel of Sonakshi at NEET Biology World. Today's topic of discussion is Hardy-Weinberg principle. It is the topic from the chapter Evolution. And this topic will have some numericals also. And for that, you have to understand the topic and the concept should be crystal clear. So let's discuss first Hardy-Weinberg principle. So Hardy-Weinberg principle states that the genetic equilibrium. So it is the principle of genetic equilibrium. It states that gene frequency. So it states that gene frequency of a randomly mating population is constant and it is stable and it is 1. So for a randomly mating population, gene frequency is 1 and it is stable. If there is any changes of gene frequency, then there is a chance of speciation. Speciation means there will be development of new species. I mean, during the course of evolution, if we find out the reason behind the evolution, then the changes of gene frequency may be the one of them. And it is proposed by Hardy and Weinberg. Now let's discuss about what is gene frequency. We know that gene will occur as allele. Means in our homologous chromosome, there will be alternate forms of a gene. And that gene will be, or that genes or the alternate forms of genes will be called as alleles. So let's say this is a homologous chromosome. And in the homologous chromosome, if in this position A gene is present, capital A, and in the same location there may be small a. So in each and every individual, we will find one pair of allele for a particular gene. Now, let's say, now we have to understand about gene frequency. What is gene frequency? Gene frequency is the total or the number of specific allele divided by total number of allele. So, gene frequency, GF, equals to number of specific allele in a population. Population means group of individual by total number of allele. Now how we can interpret? Let's discuss. For example, let's say there are 5 individuals in a population. Individual 1, 2, 3, 4, 5. So in each individual, there will be 2 alleles as I told you. Let's say the name of the allele is capital A and small a. And the combination may vary. Means, let's say any random combination you can guess. Suppose in this individual, the combination is capital A, small a. Here it is capital A, capital A. Here it is small a, small a. For individual 4, it may be capital A, small a. And for individual 5, you can have any kind of combination. Capital A, small a. Now, what is the gene frequency? As I told, number of specific allele by total number of allele, right? So here, gene frequency, here how many alleles are there? Total alleles in this population, which will have the 5 individuals. So each individual will have 2 alleles. So 5 individual will have 10 alleles. So total number of alleles will be 10. And what is the frequency of small a? So we can write gene frequency of small a, or let's say it is capital A. So how many capital A is present? 1, 2, 3, 4 and 5. So 5 by 10. What is the gene frequency of small a? Again, it will be 5 by 10, right? Now, here 5 by 10 means 1 by 2, or we can say 0.5. And here it will be 0.5. So, if the gene frequency of A is denoted as P and gene frequency of small a is denoted by Q, P plus Q will be 1. 0.5 plus 0.5 will be 1, and it will be 1 for a randomly mating population. If there is selective breeding, this constant will change. Now so it will be constant for a randomly mating population, and there will be no chances of evolution if it is randomly mating population. But if there is changes in gene frequency, what is the reason behind that? I will discuss. If there is any changes, for example, let's say this A become A prime, any other gene. So the frequency of A after the change will be 4 by 10. So it will be 0.4. And if we calculate the frequency of A, capital A plus frequency of small a, it will be 0.5 plus 0.4 after the changes. So it will be 0.9. It will not be 1. So when there is any changes of gene frequency, that can lead to the speciation and evolution. I mean, there is a change in gene frequency, right? So this is the concept of frequency. Please, if you understand this concept, any question, percentage, gen, individual, heterozygous, homozygous, everything you can do it. Now, Hardy-Weinberg states that if P plus Q equals to 1, then they told that P plus Q whole square equals to be 1. Now, if we expand that, we all know P square plus 2PQ plus Q square will be 1. And what they told that P square denotes frequency of homozygous dominant, and 2PQ means, or it denotes the heterozygous, or the number of individual or the frequency of heterozygous allele, and Q square denotes frequency of homozygous recessive. So this P plus Q equals to 1, and P square plus Q square, P plus Q whole square will also be 1. So that is nothing but genetic equilibrium. I hope you understood that. Now, some of the factors are there which can change the genetic frequency. What are those? We will discuss that. Okay. We will discuss that. Let's say, or there are factors factors affects the genetic equilibrium. What are those factors? First factor is gene migration. I will discuss one by one. Gene migration, or it is also called gene flow. Second one is genetic drift. Third one is gene recombination. Fourth one is mutation. And fifth one is natural selection. Now let us understand. Now what is gene migration? Suppose a population, when it is fragmented, means some of the individual of this population migrated from one place to another place. So what will happen? Some of the gene which is present in the original population, it might not be present in the new population. So at the new place, when this population will grow, it may lack some of the gene, and that can change the genetic frequency. So it will occur. So as the name suggests, gene migration means when individuals of population of a particular population migrates. And what is gene flow? If the migration happens multiple times, then there is a high chance of changing of genetic frequency. So gene flow means gene migration when happens multiple times. Okay, then that is called gene flow. When the gene flow, or we can say gene migration or genetic frequency change or disturbance of genetic equilibrium will occur by chance, that is called genetic drift. So when gene or genetic frequency changes due to by chance. Chance means, for example, by natural calamities, for example, by epidemics. We cannot control that, right? So if it is happened by chance, then it will be called as genetic drift. Coming to gene recombination, we know that new combination of gene will be formed during the meiosis, during gametogenesis, right? There will be crossing over. If we know the cell division chapter, then we know that there will be crossing over, and then there will be formation of new set of genes, and that is called gene recombination. So gene recombination means new combination of genes formed due to crossing over. Due to crossing over during gametogenesis. All this will form new set of genes, and that can change the genetic frequency. Coming to mutation, All we know that mutation means there is a sudden change in the basis, right, of DNA. If it will happen, then it is called mutation. And mutation, most of the time it will be lethal, means the organism will not survive. But some of the mutation are not lethal. The organism can survive. If it survives, then there is a change of that particular gene to appear a new gene, right? So mutation, it is the changes of DNA by changes of composition of DNA, or we can say DNA. There are different types of mutation by mutagens. Coming to natural selection, we know that Darwin proposed the theory of natural selection. Means if there is, or if there is any environmental changes, the organisms which will have the characteristic to adapt in the new environment, they will be selected. Others will not be selected. That is called natural selection. And the selection procedure, which is done by nature, that can change, or that can select some of the gene. Some of the gene may not be selected. So that's why there is a chance of the changes of genetic frequency. So natural selection, all we know that selection of favorable species, right? So there are 5 factors which can change the genetic frequency. Means from 1, it will vary. We know that P plus Q equals to 1. If there is any changes of genetic frequency, it will not be 1. It will be other than 1. Okay. So what are the factors? First, gene migration. If it is occur by chance, it is called genetic drift. Genetic recombination, new forms of genes or combination will form during gametogenesis. It can change the genetic frequency. Mutation, changes of DNA, and natural selection, which is proposed by Darwin. Now let's understand, or in the next video, we will discuss about some questions. My request to you that you should understand the frequency, okay, the concept of frequency which I discussed. If you understand that, whatever question will come, I will discuss the PYQ as well as new questions. Whatever questions will come, you can attempt, okay, you can attempt. And conceptually, it should be very, very clear. That's why I discussed about the genetic frequency, okay. So thank you for watching the video. See you in the next video. If you feel that this content is helpful, please like, subscribe and share my channel. Okay. Thank you. Thank you so much, dear all my students.